This is going to be a video about the difference between mitosis and meiosis. And like we all start off, let's start off with our cell. So I'm drawing our typical cell here. Here's the cell membrane. Let's give it a nucleus. Here's the nucleus. And now within that nucleus, there are going to be some chromosomes here. So in this particular case, I'm drawing one big chromosome from Dad. Here's Dad's big chromosome, and it's going to be one little chromosome from Dad. Because it's a eukaryotic organism, it's got to be a matched pair. So there's got to be a big chromosome from Mom and a little chromosome from Mom. So in our particular cell, my diploid number is 4 because I got 1, 2, 3, 4 chromosomes. Because it's eukaryote, they are arranged in pairs. I've got pair number 1 here and pair number 2 here. And these two chromosomes, they're homologous, meaning that they've got the same genes on them, but the genes may specify different conditions of that gene, but that's okay. Now let's look at the different ways they can reproduce themselves. So what we're going to look at first is going to be the process of mitosis. And the purpose of mitosis is to make more cells. In other words, at the end of this process, I want to have a replica, 2 cells that are just like this one in every way. I just need to have more of these cells here. So let's see how that process is gonna happen. So let's clear our workspace here so we can see a little bit better here. And I'm just going to draw the chromosomes here. So just looking at the chromosomes here, I've got my big chromosome from Dad, my little chromosome from Dad. I've got my big chromosome from Mom, my little chromosome from Mom. And so what I want to do is I want to replicate these chromosomes. I've got to replicate the DNA first. So I'm just going to do this, and I'm going to replicate those chromosomes. And now I've got a pair of chromosomes here. So I've got an exact replica of Dad's chromosome, exact replica of Mom's chromosomes, and they're paired together here. Now, these two chromosomes are going to be sister chromatids, meaning that they're absolutely identical. They've got the exact same genes in the exact same places. Everything is exactly identical to them. And now, in the process of mitosis, how these things are going to divide, mitosis just has one division. So in one division, I'm going to have to line up these chromosomes. Now, chromosome is going to divide horizontally here, so I'm going to line them up in the horizontal plane. So I'm just going to move these things around a bit so they can show you how they line up. They line up this way. We're actually making the arrangement of chromosomes as they would be on the metaphase plate here. So they're lining up, and this is called lining up independently, meaning as long as they're lining up in a line here, then they're fine. They don't really care in what order they are. They just need to be on the metaphase plate line. And now, when the spindle fibers come down, they're going to come down right down here. You're going to split this thing right down the middle. And as they split it down the middle, they're going to draw this chromosome apart here. And now what we have is we're going to have a cell that has our chromosome count like we had before. In other words, we've still got our normal diploid number, 1, 2, 3, 4, an exact replica of what we had in our previous cell. So that's going to be mitosis. That's how that process is going to work. So let us now consider the second kind of division, and that's going to be meiosis. And in meiosis, the object is to make germ cells. In other words, to make sperm and egg. And the problem here is that the diploid cell has, in our case, 4 chromosomes. If for human, it'd be 46 chromosomes. But in order to make the germ cells, you want half the number of chromosomes. You've got to figure out some way to reduce that diploid number into a haploid number. You just can't take random chromosomes. You've got to figure out a way to make sure that your haploid germ cell has exactly one member of every pair in your nucleus here. So again, let's clear the way here, and let's look at make some chromosomes here. So we're going to have the same chromosomes here, with the big chromosome from dad, and we're going to have the little chromosome from dad. We have the big chromosome from mom, and we have the little chromosome from mom. And just like we did before, we're going to replicate these chromosomes here. So I make an exact replica here, and now I have my sister chromatids here. Exactly same process as happened before. But now the difference happens. We're still going to separate the cell in this lateral direction here. But now, the way the chromosomes line up, they do not line up independent. They are dependent. They don't line up in single file. They line up in double file here. So they're lining up this way. And in the first meiotic division, spindle fibers come here. They drag onto these chromosomes. And now, the division pulls apart the sister chromatids. So the sister chromatids are now coming apart here. And now sister chromatids come apart. So now these are separate cells. So this is at the end of the first meiotic cell division. I've got sister chromatids. They're not separated yet. Now there's going to be a second cell division. So notice how this process has been set up here. In other words, I can make a cell around it. These are actually two separate cells now. Now this process has been set up all by the way in which those chromosomes were lined up on the metaphase plate. So they are lined up double file instead of single file. They are dependent. The two number one chromosome sister chromatids must be next to each other. So when they're pulled apart, I've got both chromosomes represented here, although they're still sister chromatids. Now in the second meiotic division here, what's going to happen is that spindle fibers come down, and now there's no more replication of DNA here. These things are pulled apart this way, and in this case, this is pulled apart this way, and now I've got 4 cells here. So here is 1 cell, 2 cell, and so on. And now each one of these cells is going to represent a gamete. And this process of meiosis is going to be the same to make both the sperm and the egg. It has half the chromosome count. So this is a haploid cell. Instead of having the 4 chromosomes like we had before, it's only got 2. But it's got 1 representative. Every cell has 1 representative from the big chromosome and from the little chromosome. And so when they come together, when the sperm and egg come together, I'm going to have a matched pair of big chromosomes and a matched pair of little chromosomes. So that's going to be the process of meiosis. So what you have to remember is everything depends on how those chromosomes line up at the metaphase plate during that first division. If the lineup is independent, they're all lining up single file, then it's going to be mitosis. You're going to make an exact replica. If it's dependent, whether the sister chromatids of the cells line up next to each other, so they're being pulled apart without separating the sister chromatids, then it's going to be meiosis.